So this video describes the lab determining the percent composition of an aqueous hydrogen peroxide solution. So generally the lab is, you're going to do the catalyzed decomposition of hydrogen peroxide by yeast. So you're going to take hydrogen peroxide just from a retail bottle, and you're going to add baker's yeast. Baker's yeast has an enzyme catalase which can decompose hydrogen peroxide. It takes 2 moles of hydrogen peroxide, converts it into 2 moles of water and a mole of O2. So you're going to measure the amount of oxygen that's produced to then back calculate using stoichiometry to figure out how much hydrogen peroxide was in that solution. Where you're going to measure using a gas collecting apparatus that will basically, you'll be able to measure the volume of gas that's displaced in this reaction using water, a water-based measuring device. This lab is pretty complicated, but it does have just a few basic objectives that are covered. Mainly you're going to, it's a quantitative lab and with some sophisticated glassware, and you're still doing a chemical reaction. Enzymes are very important catalysts involved in a lot of chemical and biochemical, especially biochemical processes. They basically are the fundamental basis for biochemistry and for life. And you're going to be doing, we're not really going to go into enzymes too much in this one, but being able to run those is a good example. And you know, I think if you do this experiment carefully, you can get really good data and really manipulate the gas law and see how all these components sort of are used with the gas law in a more real-world application. Now, of all the labs, this lab has the most changes compared to the manual. So we're actually, and that's because we have a different collection device for the gas. So there's actually a separate set of instructions and protocols. So still read the introduction, how to do the calculations. That's going to be essential in actually getting your problem correctly, but the protocol itself and how to use the device will be provided separately. So in addition, we're going to double the amount of hydrogen peroxide. We're going to use 10 milliliters. We're going to use a graduated cylinder instead of volumetric pipette to measure that out. And I think we only really need to run 1 trial. You'll have to check with your instructor. There are certain numbers you sort of expect this to produce, and if you're off because of the system's not quite set up right, then maybe you can repeat it. But I think 1 trial is usually sufficient for this thing to get good data that you can work with. Relatively speaking, this is a fairly safe lab. We're using commercial hydrogen peroxide, so you know it's mild irritant. I don't want to get it on you too much, but it's weak enough that it'll wash off with water fairly easily. We are using some sharp sticks. You might poke yourself with a stick. Broken glass, things like that. So you use the normal precautions, just you're going to need goggles and all those sorts of things because of the environment that you're working in. So our apparatus is in the manual, but you can see it's sort of this Florence flask, and it has, so we want to fill that with water and put the clamp on it as you can see here. And so this will be what will be displaced then to measure the oxygen. Okay, so this will all be on your bench. So there is a stopper, so it has 2 valves, one that's long and one that's short, and so you just need to get this set up. Now the first thing you need to do is get a siphon going. So you can either push it or pull it or whatever, but you need to get a siphon going from the Florence flask into a beaker. Okay, now we want to make sure that the water pressure doesn't add any sort of additional pressure. So what you should see if the siphon's active, you should see how the water level goes up and down. So you can see how it goes up and down here when we move it. Now this is all detailed in the supplemental protocol, but what you'll want to do is you'll want to get those levels equal, okay, and then there's a little pinch clamp that will allow you to pinch off this the tubing to this. And so the idea here is we don't want the water pressure, differences in heights of the water pressure that contribute to the amount of material and the pressure that's put off. Okay, so we need to have those equal. So there'll be a couple different places, it's well spelled out in the protocol, where you need to level the thing. And so this is one of them. So get it level, don't break the siphon, keep it underneath the water the whole time, and it should be pretty straightforward. Now at this point you're going to measure out 10 mils of hydrogen peroxide, and it's not important that you get exactly 10 mils. The main thing is that you know exactly how much you have. Okay, and then we're going to transfer this to a large tube, and this is one that'll be given to you. It'll be part of the device, and I would we really want to make sure it's at the bottom of the tube. So I would use a pipette to make sure it gets all transferred. We can clamp and put the stopper in. Now, what we're going to do is pull the little stopper part off and just leave that out, so then we can get our stick ready. So the stick, we're going to put a little Vaseline on the whole thing. These are just wooden sticks. So you need to sort of lube the bottom 2/3 of it with Vaseline. Okay, and that'll allow you, because this is going to slide up and down through this stopper to put the yeast into the tube. And so you need to first make sure that your stick can slide back and forth, and then put a little extra glop of Vaseline on the bottom. So now you can see it's sliding pretty nicely, and we're putting on a little extra glop. And then you just don't need a lot of yeast, just a little bit of yeast. So it'll be in a watch glass, just pick it up. And the key thing is it has to be, it can't be loose. You've got to make sure there's no loose yeast, because that'll ruin your experiment if that falls off too early. And finally, you can reattach the tube, the hose to that, and stick that into your tube, being careful not to lose any yeast. And you don't want the yeast to be in there quite yet. Okay, so now this part is set up pretty well. And we have a closed system now from the gas collection. So now we can open up the clamp, and they're not leaking, and we should be ready to go. Okay, so when we're ready to do the experiment, we switch to a dry beaker. Okay, so one that doesn't have any water in it, and we stick the yeast in. And so you should be able to see some vigorous bubbling from the yeast. It'll take, about 10 or 15 minutes. You can see it's pretty foamy, so you're going to need to sort of beat on it to get the glass out of there. But at the same time, it's going to start displacing water. So you will see the water then being displaced into the flask. When it's done, you'll want to measure the temperature of the water. That'll be important. We're also going to measure the barometric pressure eventually. Okay, and then when we're done finally, when you think it's done, sort of hit on it, make sure there's no foam left in there, pull the tube back out to its original location, and then again do the equilibration of the levels, of the water levels. So you can now measure the amount of water that was displaced in the graduated cylinder. 100 ml will be enough usually, I think, so that will allow you to get the amount of gas displaced that way. So that's part of the gas calculation. All right, so we're going to work in pairs. Again, don't put on too much yeast. You only need a few. It's a very vigorous reaction. And then make sure the foam is sort of broken up before you stop measuring everything. If you have to tap it or do it a little shake, anything where you can still maintain the seal of the reaction, and the siphon, if you have siphon going, keep the siphon going. Keep that seal intact, but try to get rid of as many bubbles as you can. Now, there are certain numbers we just don't have uncertainty on, and those are conversion factors. Okay, if we know that 1 atmosphere is 760 torts, it's exact. It's not got an uncertainty at all. And the same is with stoichiometry. So if we're talking about doing a stoichiometric ratio, which we'll have to do in this reaction because we use 2 mole, we produce 1 mole of oxygen for every 2 moles of hydrogen peroxide consumed, we know that that also is exact. So we don't have to add uncertainty there. So the uncertainty then needs to come in with our measurements, things like mass, volume, all those sorts of things.